Subcultural Characteristics Lab. In this lab, we will study the growth characteristics of certain bacteria on different media. You can see these media pictured on the right side: the Auger Plate, the Auger Deep, the liquid broth, and the Agar Slant. Now, when we study characteristics, we look at this picture with colonies and we notice that they have different characteristics. They have different size, different color, different borders. If you're wondering what a colony is, you can look on the right side, and those little tiny dots of bacterial communities are colonies on these plates. Another example, we will notice the different growth patterns of bacteria on these Auger slants. Some will grow more like big beaded colonies, some will have smaller beaded colonies. The bacteria used in this lab are the following: Micrococcus luteus, growing in a liquid medium. Pseudomonas aeruginosa, also growing on a liquid medium. Mycobacterium smegmatis, in liquid. Bacillus cereus, in liquid. And notice all of these are labeled from our lab staff. And E. coli, the last bacterium, also in liquid. Notice not to pick up ever the test tube from the white cap, but support it from the bottom. You don't want it to fall and then you create broken glass and also have a bacterial spill. So very careful with the white caps. Now the sterile media that we're going to use for subculturing will be a sterile Agar Slant. You can see it's slanted. This is a solid agar that carries nutrients for bacteria to grow on the surface. Each bacterium will have one of these agar slants. We have a sterile liquid broth. You can see it's liquid when we slightly tilt it. We will also put each bacterium into a liquid broth. This is also called subculturing, if you remember, when we transfer bacteria into media. This is a sterile Auger plate. Each bacterium will have its own plate. And we also have a gelatin tube, NG, sterile here, but we will subculture each bacterium in this solid gelatin tube. Now, the thing to note is that gelatin is used to detect bacteria that have an enzyme called gelatinase. Now, gelatinase is an enzyme that breaks down gelatin. Gelatin is a product of collagen. Not all bacteria break down gelatin. Other tools we are going to use include a loop and a needle. These are called inoculating tools. We will use the loop to subculture or inoculate our agar plates and our liquid broths. We will use our needle to inoculate, subculture our gelatin tubes and our slants. Now, we also have a nice hot baccinerator with a switch, turns on and off. It's ready to go for our lab. Another little item that your instructor may be using is a disposable loop. If you have these in the lab, you need to make sure when you use them, you dispose of them in the hazard trash. Another little note to make is that some instructors like the Bunsen burner flame instead of the baccinerator. And these we use to sterilize our equipment. Moving on to the technique, don't forget our PPE, gloves, goggles, and lab coat. We will be doing labeling. So we will be picking up our Sharpie and labeling a plate here with one of the bacterial names, Bacillus cereus. I don't want to be in the middle when I label because that will interfere with reading my results. I want to stay on the perimeter, or the periphery as we say, of the plate out here. I will also label an Agar Slant for Bacillus cereus, and I will also label a gelatin for my Bacillus. I also will label a liquid for my Bacillus cereus. So you can see that my Bacillus was labeled on 4 different media. Now here's Pseudomonas aeruginosa. I want to make sure I've labeled a Pseudomonas aeruginosa Auger Slant. I want to make sure I have a Pseudomonas aeruginosa labeled nutrient gelatin. Want to make sure that we have a liquid labeled Pseudomonas, and of course an Agar Plate labeled Pseudomonas aeruginosa. We repeat this labeling for all the remaining bacteria. So now we are ready to move on to subculturing, transferring bacteria from the original to the new sterile media, the 4 media, the Agar Slant, the gelatin, the liquid, and the Auger Plate. We place our Pseudomonas nicely organized on a rack, and then we want to make sure we have everything organized and ready to go before we start subculturing. We want a nutrient gelatin, a liquid, the Auger Slant that is labeled Pseudomonas, and we also want to pull out the plate that we've labeled Pseudomonas. Getting a good system down after labeling is very important in order to go through the lab. Here's our loop. We will be burning the loop, sterilizing it, in the baccinerator. This is called the aseptic technique, which means that it will not allow for contamination. We burn for about 10 to 15 seconds. The wise thing to do in order to go through the lab a little faster is to sterilize maybe 2 or 3 loops, and that way while one is cooling you can be using the other one. And you'll see we are going to sterilize a 3rd loop here for our lab. You also notice we are placing our inoculating loops on the side of the baccinerator, and here we're lifting it so you can see there's little tiny pockets where you can set your inoculating tools after you have sterilized them. So now we are ready to move on to the actual technique. We pick up our Pseudomonas aeruginosa, use our ring finger and our little finger to pull off the white cap, go in with the loop into the actual original Pseudomonas aeruginosa, best to go to the bottom to collect a good sample. And now we will transfer this Pseudomonas sample or subculture it using the aseptic no contamination technique into a new sterile liquid. We go in and we swirl around the new Pseudomonas culture. So now it's no longer sterile. It carries bacteria in it. We will sterilize our loop. Pick up another loop that's been cooled. Want to make sure those loops are cooled so you don't kill the bacteria. Use the ring finger, the little finger again. Go in, get some more Pseudomonas sample. Come out carefully, cap the tube, place it aside. And now we will pick up the plate. With this plate we will be doing a 3-way streak plate. You'll notice we'll zigzag back and forth, skate on this nice auger, but not to pierce it until we reach a certain point. We will sterilize our loop. We will turn our plate 90 degrees. And after the loop has cooled, we will make a 2nd streak. We will drag bacteria from the first streak over into an empty space and do a 2nd streak. This allows for the bacteria to get fewer and fewer as they grow over 2 days, and our goal is to eventually, after a 3rd streak, to get colonies. You hear the 2nd streak, zigzag and we stop at a certain point. We turn the plate 90 degrees and we sterilize the loop for about 10 seconds. Want to make sure we cool the loop. We do not pick up any new bacteria between streaks. So now we will drag our second streak over and make a third streak into the empty space. It is very important for us to follow this procedure in order to get good colonies to study for the characteristic results. And there's our third streak into the empty space. We sterilize our loop. And we are now completed the auger plate. Now, the auger plate here is out in the open because the instructor has to show you how the streaking of the plate is. But you want to make sure you know where the lid is. The bottom part is the part that has the actual auger. And then we label the back, the bottom as we say, not the lid because the lid can fall off or it can be misplaced. So really when we inoculate these, we place them on the table and barely lift the lid to make the streaks. But again, in this video, we had to have it nice and open so you can view the streaking method of the plate. Now we will use the needle. We sterilize our needle. And again, if you'd like to go a little faster, you want to sterilize a couple needles and have them ready. So now we are ready, after the needle cools, to pick up some more Pseudomonas aeruginosa from the original culture. Very careful not to let the white cap touch anything because then we can get contamination introduced. We go to the bottom and we pick up bacteria. Now some people think we didn't pick up enough, but there's plenty of bacteria on that needle. We pick up our sterile gelatin. We uncap it. And then we go in and stab in the center the gelatin about 3/4 of the way down. We don't want to go all the way down. We don't want to introduce air into the bottom of the test tube. We want bacteria that grow on the bottom with a little bit of oxygen to be able to use their gelatinase and metabolize gelatin if they have gelatinase, and the ones on the top that use oxygen to be able to metabolize gelatin as well. We sterilize our needle. We pick up another cool needle. And now we will go into the Pseudomonas aeruginosa original culture again with a cool loop needle. And we will now inoculate subculture, the Pseudomonas, into this Auger Slant. Now notice with the Auger Slant, we have to go into the bottom of the slant. We don't want to pierce the slant, but we want to skate on the surface and zigzag out. Literally draw a zigzag and come all the way out. Go to the bottom and zigzag out. And now our Auger Slant is complete. We sterilize our needle. And now we are ready for the next bacterium. We pretty much follow the same procedure for all the rest of the bacteria. We want to make sure we keep our tubes and plates organized. And we will now pick up Mycobacterium smegmatis and subculture Mycobacterium. We organize the gelatin that we've labeled for Mycobacterium, the liquid, the slant, and the plate. We will pick up a loop that has been sterilized and cooled, go to the bottom of the Mycobacterium smegmatis culture, pick up our liquid, Go in and swirl around. So now we have subcultured mycobacterium into this liquid. We burn the loop, but we have another loop that's been cooling, so we want to pick that one up. We will pick up new Mycobacterium smegmatis from the bottom. You can see it's a little clumpy. Some of them create the sediment on the bottom. And now we will streak our plate three-way. We zigzag almost halfway through the plate. We sterilize our loop. We turn our plate 90 degrees, and with a cool loop, without picking up any more mycobacterium, we drag the first streak over into the empty space, making a second streak. Turn 90 degrees, sterilize our loop, let it cool, and now we are ready to drag bacteria from the second streak into the empty space, making a third streak. The plate is now complete. Now we will use the needle. We pick up some Mycobacterium smegmatis. Uncap our gelatin. Stab our gelatin halfway, 3/4 of the way in the center. Sterilize our loop. And now we are ready to pick up some Mycobacterium and subculture it into the Auger Slant. Again, we go to the bottom of the slant, we touch the surface, we don't pierce the auger, and we skate zigzag out of the auger slant, touching the surface only. Sterilize our needle. And now we have completed the subculturing of Mycobacterium smegmatis. We move on to our third bacterium and repeat the process. We have Micrococcus luteus. We organize our materials that we will need to subculture. And now we are ready to start subculturing Micrococcus luteus. We will use a loop. We will pick up some Micrococcus luteus. Again, we want to go to the bottom and get a good concentrated sample. Open our liquid and swirl it around. Sterilize our loop. Go into the Micrococcus luteus again. Pick up a sample. Open our plate and do a three-way streak plate again. Here's our first streak. Sterilize our loop. Turn the plate 90 degrees. Wait for the loop to cool. drag bacteria from the first streak over to the second streak, turn our plate again and sterilize our loop. Cool the loop. And now we will drag bacteria from the second streak into the empty space, and that will be our third streak for the plate. Sometimes we have to play with the light to see where the second streak is, and there we go. That's the third streak. We will now move on to the inoculating needle. We will pick up Micrococcus. We will stab our gelatin in the center, 3/4 of the way down. Sterilize our needle. Pick up some more Micrococcus luteus. And now we will inoculate subculture the Micrococcus into our Auger Slant. Going in, touching the surface and zigzagging out. And that completes the subculturing of Micrococcus luteus. We organize our materials to make space for the other two bacteria that we have to subculture. Now we will move on to a different method. We will use, instead of the metal loops, these disposable loops. You can see a loop on one end and a needle on the other end. They come in little plastic bags. They are sterile. And we push the needle end out. We never touch the needle or the loop side. We touch the handle in order to make sure that we do not contaminate the two ends. So instead of the metal loop now, I will demonstrate the use of the plastic loop, the disposable loop, for subculturing Bacillus cereus. We go into the culture, get a sample, pick up our Bacillus labeled liquid, go in and swirl around. Now this is not metal, so there's no way we can put it in the baccinerator and sterilize it. We have to put it in the hazard trash. Now if you'd like to save loops and not waste them, you notice there's a needle, and if I'm very careful and I turn that other end, very careful not to get any bacteria on my gloves from the loop end, I can dip the needle end into the Bacillus cereus, and I can go ahead and stab my gelatin deep. Now notice this is a little bent, so it's not going to quite be a straight, going in technique. That's okay. It was a little bent there. And now we are ready to toss our disposable loop and needle into the hazard trash. We pick up another disposable loop. We pick up our Bacillus cereus, dip in to get a sample, and we will now do the plate. Our first streak, we cannot sterilize this, so we do have to throw it away. We have to get a second loop for our second streak. Remember, we will drag bacteria from the first streak into an empty space to form that second streak from the first streak. Well, we can't use this loop anymore. We have to toss it. And now we will have to get another one out. We have to tear these apart, make it a little easier on us. And now we are ready to do our third streak. We play with the light to see where our second streak is. Turn 90 degrees and we take over some bacteria from that second streak into the empty space. Now, I can use the needle end before I throw this away, so I might as well use it. Again, it's a little bent. So we go into the Bacillus cereus. We open the Auger Slant. We go in and we zigzag on the surface of the Auger Slant. It's a little difficult here, you can see, because it's bent. If this happens, usually you can fix it before you take it out of the bag. And we throw away our loop. So we have completed now Bacillus cereus subculturing. We'll organize everything onto another rack. And finally, we have E. coli to subculture. We will use disposable loops again so you can see this other method besides the metal loop. We will pick up a disposable loop, pick up some E. coli, carefully never to fling the loops or the needles. Go in, swirl it around, and now we have E. coli inoculated into that tube. We can use the needle end. Go in very carefully and stab our gelatin. This is a little straighter for the needle. Yeah, you can see it going straight in. That's perfect. And then we throw away our disposable loop. We are almost done with demonstrating the technique. We use another disposable loop, dip into the E. coli, take our plate, do our first streak, throw away our disposable loop, turn 90 degrees, use a clean loop. From that first streak, drag bacteria over to a second streak into an empty space, turn the plate 90 degrees, and this will be our third streak. And there it is. And that completes the Agar plate for the E. coli. We can still use the needle. So now we will take the needle, dip it into E. coli, take our Auger Slant, go to the bottom, and touch the surface and skate on it, zigzag out. We throw away our disposable loop. And now we have completed the lab. There are two more steps that we need to take. We need to first make sure we take out the original stock bacteria, the ones that were given to us. And then place the new bacteria, the new media, into an incubator, 37 degrees Celsius or 98.6 degrees Fahrenheit. Place them in the incubator for 24 to 48 hours and come back and read the results. Please do not forget, the next step is to clean up. Want to make sure we keep everything clean, throw away our trash in the trash can. Here's the trash can. We want to turn off our Bexinerator. Make sure our hazard trash goes where the instructor has instructed us. Usually it's placed in an autoclave either the same day or the next day to destroy the waste. We want to make sure our original bacteria are given to the instructor. We put plenty of bleach on our table and use paper towels to disinfect our area. Very important to clean the lab when we are done. We can throw this in the trash can since it does not have bacteria on it. Our loops and needles need to be disinfected. Anything that was contaminated, if it was your gloves or paper towels that had bacteria, you need to throw them in the hazard trash. Make sure you take off your gloves as shown here. These gloves, I had no spill on my gloves. I don't have any bacteria, so I can throw them in the regular trash. We want to wash our hands, come back in a couple of days, read the results, and do our lab report. Thank you.